So this video is going to discuss a technique that is very powerful and used by many scientists to study the function of proteins. This technique is known by a number of different names, either RNAi, standing for RNAi inhibition, or siRNA, using small inhibitory RNA molecules, or sometimes shRNA, short hairpin RNA. Anytime you see these terms, you're going to be looking at an experiment that is going to perform a knockdown experiment. And let me explain what scientists would use this for. So here's a cell, and let's say there are proteins X, Y, and Z in the cell. And as a scientist, you want to study these proteins, their functions, their interactions, and the theory that you might have is that when the cell gets some signal, that protein Z affects the function of protein X affects the function of protein Y. And so how could you prove this? How can you study this process? One way is to, if you could, manipulate the cell such that you can get rid of protein Z, erase it from the cell. And then when you treat cells with whatever you're treating them with, well, does something not happen to protein X or Y, or does it still happen? So this is known as a knockdown experiment, manipulating the cell to have a protein basically disappear in the cell. It's a very powerful technique. It actually won two scientists the Nobel Prize in 2006. They won the prize for their discovery of this process called RNAi, which is apparently a natural process that cells use to control the levels of their protein. We think about genes and promoters. It turns out cells use small inhibitory RNAis to control the levels of their proteins. And in fact, one of the researchers who won the Nobel Prize is a professor at the University of Massachusetts Medical School. All right, so how does this work? There are lots of videos online you could watch. I'm going to give you a very brief description of it. So let's say the researchers wanted to study protein Z. So here's the gene for protein Z. There's the DNA, and we know when genes are turned on, because promoters are activated, the genes transmit that information to mRNA molecules, so that's transcription. And then those mRNA molecules are translated into protein. So there you go, the cell is making protein Z. Now, if we want to manipulate the cell to get rid of the production of protein Z, we could add small inhibitory RNAs. So these are RNA molecules that are in the range of 21 to 23 nucleotides long, and scientists can genetically engineer them in the lab, chemically synthesize them, and what they would do is they would design a sequence that would match the complement of the mRNA. So this small inhibitory RNA can be added to cells, it gets into the cells, into the nucleus, or cytoplasm, and attaches to the mRNA. So it forms this double-stranded RNA molecule, this little piece. So if we're targeting the Z gene's mRNA, we would design a nucleotide stretch, 21 to 23 nucleotides, to complement something that's very specific to this mRNA and that wouldn't be present in any other mRNA. And what this small region of complementarity does is recruits proteins that destroy the mRNA. And there are lots of names for these proteins, and it's a big complex, and you don't have to really worry about that. Some of you might have learned about it in cell biology or molecular biology or genetics. But I'm just talking about what the technique is going to be used for. So now the mRNA for Z is destroyed, which means the production of Z will be diminished. Or the Z protein won't be made anymore. Even though the gene is on, the mRNA is erased, the protein is not made. So this is called RNAi knockdown. We have knocked down the production of a protein. And so when these experiments are performed, you'll see, for example, in a paper, it might say SIZ. And that would mean the researchers have added a small inhibitory RNA to knock out and target the Z gene, specifically the mRNA made from the gene. When you read papers, you'll see different ways that researchers write terminology. So they might write SIZ, they might write SHZ, because instead of small inhibitory RNAs, sometimes researchers add short hairpin RNAs, which get into the cell and also attack the mRNA. Something else you'll commonly see are control RNAs. So small inhibitory control RNAs, sometimes called scrambled RNAs, they're sent into the cell. They're not going to target the gene. So they're randomly put together nucleotides that don't target the gene. And so those would be the negative control. So let me give you an example of what an experiment would look like. So let's say we're going to do Western blot for protein Z, and that's the normal amount of protein Z in a cell. If we added SIZ or SHZ, that would be short inhibitory RNAs that target the mRNA of protein Z, or short hairpins. It's a variety of ways to target the mRNA. Either way, adding that to the cell, you see caused a greatly decreased amount of protein Z to be produced. So it's diminished, it's knocked down. Sometimes it goes away completely, sometimes it doesn't, but that's okay. There's much less protein Z in the cells analyzed in number 2. In lane 3, let's say the researchers added a control RNA, short interfering RNA. It doesn't interfere with protein Z is made, so that usually you'll see a control. And then researchers will add drugs or add growth factors, all sorts of things when they're doing these types of experiments. So again, if you were studying proteins X, Y, and Z and their interaction and their functions, you might try to knock down protein Z and say, okay, well now how does the cell work? Does it grow? Does it not grow? Does it signal? Does it not signal? So there are a wide variety of experiments you'll see RNA knockdown performed in. I do have one example I pulled off the internet. So here's a figure using some proteins that we know. You know the EGF receptor, you know HER2, which can form a heterodimer with the EGF receptor. TS, well, that's some protein, I don't know what it is, but that's okay. And alpha-tubulin, that's a very common loading control. So sometimes you'll see actin, sometimes you'll see tubulin. It's just a protein that's abundant in the cell, and the levels don't really change, shouldn't really change. So if you look in lane number 1, they're using the SI control. So they're adding small inhibitory RNA to the cell, but it's not really targeting any genes. It's a random assortment of nucleotides, and there's a certain level of the EGFR protein, you can see there by Western blot, and the HER2 protein, the TS protein, and the alpha-tubulin protein. In lane 2, they add a small inhibitory RNA that targets the EGF receptor. And what do you see? Well, you see the production of the EGF receptor dramatically decrease. So the production has been knocked down. If you look at the other proteins, though, HER2, not affected, because this small inhibitory RNA is only targeting the mRNA that comes out of the EGFR gene. So all the other proteins, not affected. In lane 3, they're adding small inhibitory RNAs that target the HER2 gene. And if you look, there's the production of the HER2 gene. It has been knocked down. EGFR, plenty of EGFR. That gene is unaffected. And tubulin should also be unaffected. In lane 4, they add a mixture. And so they've knocked down both genes. So you can see EGFR levels have decreased and HER2 levels have decreased. Now this is interesting because whatever TS is, its levels seem to have gone down as well. And that's not an direct effect of the siRNA probably, but maybe it's an effect of when those two proteins are absent in the cell, whatever levels of whatever TS is, its levels might go down. So that's an interesting experiment. So I want you to know this technique, RNAi, because you will see it in papers that we will read in the future. Yes.